Okay, so part 2 of chapter 5 is going to cover membrane transport. So as it relates to our membrane, we have to ask ourselves, what do we want to come through the membrane, right? There's going to be a lot of ions and a lot of molecules that can come across it, but not every single thing should come through it. And because of that, our membrane is known to be selective, or to have selective permeability, meaning that it's selective, right? It can pick and choose what actually can go through, and permeate means to literally go through. So you may have heard of something like it may permeate a fabric, like a pen may permeate a fabric. That means it has the ability to go through that fabric. So selective, choosing permeability, what to go through, is exactly what our membrane has the ability to do. An example of this is actually the blood-brain barrier. So your brain, of course, is the power central nervous system of your body. It's going to be the operating system, for lack of a better word. So we want to make sure that we control what can go in and out of the membrane that covers the brain. So all the capillaries, the little blood vessels, we have very, very particular barriers or particular things in place to make sure that not everything that's in your blood, like an infection, goes into your brain. So as we think about membranes and transport, we have to ask ourselves, why would an ion want to move from one place to the next? So why is it actually moving around? A great example is an elevator. So especially now, you jump on an elevator. Would you rather go in the elevator on the left that has tons of people there, or the elevator on the right? Most people would pick the elevator on the right because of what? It has a lot more space, right? This is exactly how membranes are going to be moving. They're going to tend to move from places that are concentrated to places that are less concentrated. So part of membrane transport is going to be two-sided. One is going to be passive transport, and the other one will be active. So as we're thinking of transport, I want you to always consider, is this passive transport or is this going to be active transport? And I have a lot of examples. Within passive transport, you can have simple diffusion. And you can also have another one I'll explain in a second called facilitated diffusion. So I'll explain both of those. So both types of passive transport are going to have some similarities. With simple diffusion, we're going to be moving from high to low. So we're going to be moving from an area that's very concentrated. So let's say we're talking about some space like this, to an area that is much lower concentration. Simple diffusion goes directly through the membrane. So our cell membrane, that molecule can go directly through it. And it's going to be moving to an area with more space. When it goes from an area of high to low, we call that moving with the gradient. My best example is the saying going with the flow of traffic. So if you're on 95 south, and you're going in the direction that is south, that means you're going with the other cars, right? Everyone's on the interstate. They're going in the southern direction. You're going in the southern direction. That is with the gradient. The other type of passive transport is known as facilitated diffusion, right? Facilitated diffusion is where, again, they're moving from high to low. So that is going to be a similarity between both simple and facilitated diffusion. And they're also, facilitated diffusion is also moving with the gradient. So we're still on 95 south and we're still going in the southern direction. So that's the same between the two. The difference is facilitated diffusion is going to need some type of transport or facilitator. So it's going to need some type of help. An example would be if you have a membrane that, for instance, has maybe a channel through it, right? So some type of protein that's going to be like a little hallway, maybe that ion that's using facilitated diffusion needs some type of facilitation or help to get through the membrane. It cannot go directly through the membrane like simple diffusion. It needs some type of help to be able to go through the membrane. So already in our transport, we have a passive transport versus active transport. Within passive, we have simple and facilitated. So simple diffusion, facilitated diffusion. There are some similarities between the two, right? With the gradient high to low, but they do have a big difference as far as simple going directly through the membrane and facilitated needing some type of transport or assistance. The active transport we have, you're going to see it's a little bit different. One of the major differences between that is active transport goes from low to high. So if we're talking about a space, right? Let's say we have some type of area, some type of substance that is really highly concentrated, and then you have another molecule here. Active transport is where we take the area from a low concentration and move it to a high concentration, right? That is not going to be as easy as going from high to low. It's like climbing up a slide versus going down the slide, right? Going down, that's going to be our simple diffusion, facilitated diffusion. It goes with the gradient, sliding down the slide. This is climbing up the slide. It's going to require some energy to go from low to high. Active transport, you also will need some type of transport protein. And again, like I alluded to, it's going to need energy. The form of energy in particular is going to be this ATP, and it goes against the gradient. So it's not going with the flow of traffic. This is 95 south, and you decide to come along in your car, and you're actually decide you want to go north on the 95 south highway, right? That's a problem. That's going against where everybody else is going. Everyone is going south and you're going north, right? That's going to be against the gradient. So another image that kind of hopefully puts it together a little more clearly, as you see here with diffusion, we're going from high to low. We're going down the slide. We're going down the hill. It does not require any energy. With active transport, we're going from low to high. So we're going up the hill. We're going against the gradient. We're going from low to high, and that's going to require the use of some energy. Again, just to kind of show you a picture that maybe, puts it into context a little bit better. When we're looking at simple diffusion, you see you have a high concentration of purple ions at the top. They're going directly through the membrane to a low concentration of purple ions. Here is facilitated. You still see a high concentration of blue ions, but it's facilitated, so it has to go through a channel or a protein, and it's still moving to a low concentration at the bottom. And then finally, active, you see at the top, we have a low concentration of purple. It's going through some type of channel, and it's also using that ATP energy to go to a high concentration. So again, just looking at the big picture, we're going high directly through the membrane to low, high with facilitation to low, low with facilitation and energy to high. So make sure you're very, very clear about the differences in those types of membrane transport. There are 3 factors that can affect diffusion. One of them is size. So something smaller can go through the membrane much easier. Think about, throwing a little tiny ion through here versus a big protein. The bigger protein is going to have a lot more trouble. So smaller ions, smaller molecules can travel through a lot faster. They can diffuse faster. Polarity. So something that's nonpolar can diffuse faster. Look at the inside of the membrane. All nonpolar. So if it's not polar, it can go through really, really fast because this is the zone that it needs to be in. It's comfortable here. Same thing with the charge. So anything that is not charged or non-charged, it's going to also move through faster than something that is charged. Because once it gets on the inside, it's going to kind of have a disconnect with all of the non-polar, non-charged tails that you find in the middle. So let's talk about osmosis. So osmosis is defined as the movement of water, right? So keep in mind, we're moving the water to balance a solute concentration through a membrane. So a few things to keep in mind here. This is just an image. Let's say this is our cell. In this particular image, we're looking at 90% H2O, which, if you recall, the H2O or the liquid is known as the solvent with a V. And then you have a percentage of the solute solution. So maybe in this case, it will be salt, it will be sugar, it will be whatever you want it to be. Regardless, I want you to make sure that you understand how to read this. This is 90% H2O, meaning 10% solute. So this will be an example of a 10% solution. On your exam and in the textbook, you will see items written this way, and I want you to make sure you can identify the solvent, the solute, and also the general percent concentration. If you see it this way, you may also see it this way, in which you have a circle, which that would represent the cell. And then additionally, the fluid outside will have some type of value there, letting you know the concentration of the external fluid. So let's say you got a 75% solution, then you should be able to determine that means 75% solute, right? So let's salt, I guess, or whatever it is we're talking about. And then it has to equal to 100% H2O. So you see that's going to be a 100% solution, in which a portion of it is going to be the solute. The other portion will be the H2O. You have 100%. Same thing here. 90 plus 10, 100. 90 plus 10, 100. So when you see some of these questions, if you have a 75% solution, or if you have a 92% solution, whatever it is, make sure you're mindful about it adding up to 100, and you know how to calculate solute versus H2O. So in this case, you would be looking at a 10% solute solution in a cell that's in a 10% solution. So a cell with a 10% solution in a water solution that is also 10%. So these are going to be the same. Make sure you're familiar with reading this, and we'll do a lot more practice in class. All right, so let's look at the first type of environment. So this is going to introduce you to a term called tonicity. So tonicity is going to be that makeup of H2O, how that solution looks as it relates to the concentration of solute in the solution, and also the concentrate of solute in the cell. So anything that's isotonic means we're going to have equal parts, equal solutes on the inside of the cell and on the outside. So this is 5% versus 5%. An example would be what we just looked at here, in which you have a 10% solution of a 10% mixture of a cell in a 10% solution, right? That's going to be an isotonic example. An example would be, let's say a gummy bear in a sugar solution. So let's say the concentration of sugar in the gummy bear was 5%. The concentration of sugar in the environment was 5%. It's going to be the same. In this case, water is going to equally move in and out of the cell at the same rate. This also means the cell size stays the same. So let me give you a few examples. I'm going to just kind of draw things out. So isotonic, if you look at the density, I hope you notice that the density is kind of similar. We're not counting the actual solute pieces, but there's not a big difference between how spread out those dots are in the circle and outside the circle. This will be an example of an isotonic solution. Let's just give it a value. Let's say it's 20% solution here, it's 20% solution here. What happens with the H2O? What happens with the water? Because it is the same on the inside and the outside, the water moves out and it moves in at the same rate. So that causes your cell size to stay the same. Okay? Isotonic, equal parts on the inside and outside, equal solutes, in particular solutes, right? We're talking about that solid portion, on equal parts on the inside and outside. The water is going to move in and out equally, and because of that, the cell size will stay the same. An example here is going to the doctor. If you ever need an IV, I know we have a lot of nursing majors. If you have a patient, you cannot give your patient pure water. Pure water will cause their blood to dilute itself. And because of